I think Bhaskaran sir has joined.
Just checking, sir.
He was in the waiting room.
Yes, I think he has joined.
Yes.
The meeting is live on social media also.
Thank you, Satish. Please see that the panelists are on the co-host panel. And let me formally start the session for today. It's 10:31. Will not waste a minute or more. A formal welcome to all of you. Good morning. I'm Swati from PharmaState Academy. Today, in the third session of the Pulse series, we are going to deep dive into the topic of quality risk management. This is a highly important topic, and we are going to give a special focus on revised Schedule M. So be attentive today. With risk-based inspections conducted by the regulatory authorities, this topic demands your attention, and we have today allotted more time towards the Q&A session. We are having top subject matter experts with us, and we hope a clarity would come on this topic. I'm confident that this session will provide you with actionable knowledge to enhance your organization's quality risk management. Moving ahead, let me thank the audience first for overwhelming response to the Pulse program. This is an open session, and we are also live at Facebook, LinkedIn, and YouTube channels. Regarding the next sessions, we will update you. The fourth session will be on, after this session, the fourth session will be on qualification and validation, and it will happen on 7th September. We have a lot of time in between. There would be a session also, extra session also. But those will be closed sessions. We will not be live at our social media channels, and there won't be any free participations from session 4 to session 25. You can all book the complete session package. We will announce and communicate the way to book and the topics soon. Before we start, I would like to extend our sincere gratitude to the major industry associations who have extended their unwavering support to the Pulse initiative. And the most recent one to join us is DMMA, Drug Marketing and Manufacturers Association from Gujarat. Thank you for joining the cause. We would also like to express our heartfelt thanks to the following associations: CP 𝗥𝗲𝘃𝗶𝘀𝗲𝗃, Karnataka Drugs and Pharmaceuticals Manufacturers Association, Himachal Drug Manufacturers Association, Rajasthan Pharmaceutical Manufacturers Association, Federation of MP Chamber of Commerce and Industry, the Pharmaceutical Manufacturers Association of Tamil Nadu, Drug Manufacturers Association, Indian Pharma Machinery Manufacturers Association. And we are also grateful to 𝐒𝐕𝐵 and 𝐶𝐸L for sponsoring the session today. They are the sponsors for this learning session. And my sincere thanks to the organizing committee, including Mr. Hari Jain, Dr. Sanjeet Singh Lamba, Arjun Kapoor, Sunil Ahuja, Ms. Aparajita, and Dr. Satish. So quickly, I wanted to also show you the agenda for today, what we are going to have for us. So today we are going to start with the keynote address by Chief Guest, Mr. 𝗏𝗏𝗃𝗍𝗏. Ms. Aparajita was to deliver the welcome address, but due to some urgency, she would not be there today with us. Then we will have a deep dive into the topic on quality risk management with Dr. 𝗥𝗲𝗃𝗏𝗏. And after that, we will have a deep dive into the topic with a panel, a very, very respected panel today, and then the closing remarks. And these are the supporting association I have already mentioned. We are also thankful to knowledge partner Trillium and media partner Express Pharma. And let's start now. We are starting with a disclaimer. The content provided in this session is for informational and educational purposes only. The views expressed by the speakers and panelists are their own and do not necessarily represent the views of FOPE or PharmaState Academy. Participants are encouraged to seek professional advice for specific issues related to the revised Schedule M. Let me now invite our chief guest for today, Shri Manmohan Taneja, State Drugs Controller and Licensing Authority from Haryana. Please join me in welcoming Mr. Manmohan Taneja a distinguished professional with over 30 years of experience in the pharmaceutical regulatory field. Mr. Taneja boasts an impressive career, having served as Drugs Control Officer, Assistant State Drugs Controller, and State Drug Controller since December 1, 2021. Beyond his regulatory expertise, Mr. Taneja is a recipient of numerous awards and recognitions. These include Best Drug Inspector of India in 2009, COVID Warrior Award, to name a few. He has also been nominated by Government of Haryana as a member of committee constituted for affiliation of pharmacy colleges in Haryana. Welcome, sir, and the stage belongs to you. Please kindly address the audience.
Thank you.
Welcome, Taneja ji, on behalf of FOPE as well. And we are very lucky to have you with us today.
Thank you, Jan
Thank
sir.
you.
First of all, I congratulate you for organizing such a wonderful program on the burning topic. You see, you have chosen a very good topic, quality risk management. Earlier, it was not included in any section of the schedule M. Now it has been specifically given a section. Apart from QRM, there are some other newly added topics like POQS, product quality review, complaint, etc. Sticking to the today's topic, QRM, as everybody knows, you are all the industry captains. What is the objective of quality risk management? It is to protect patients in terms of quality, safety, and efficacy. And it is the duty of both regulators as well as industry captains. The safety of the patient should be given top priority. That's why newly drafted Schedule M has been notified. You know, this QRM is not only application of GMP, besides it is product oriented. Risk is basically, it is combination of probability of occurrence of harm and severity of harm. During course of manufacture, there may be certain reactions due to environment, machinery, etc. First of all, we should know what is the risk, and it is done through the assessment, then control, review, and all. And it covers starting from R&D to pharmacovigilance, means manufacturing process, sourcing of raw material, production, then storage, and then distribution. It covers all. Although GMP covers critical requirements to build up quality in all products of a particular category of dosage form, yet a lot is left when viewed for each individual FPP. And even excipients play very vital role. You know, there are few high risk excipients like IPA, glycerol, propylene, ethylene, glycol, sorbitol, etc. And we have seen so many cases of diethylene glycol impurity in ethylene in PG and sorbitols, glycerol, etc. And even our reputation was at stake once a time, but it all settled due to the intervention of Government of India. And certain steps were taken by the ministry. Though we have lost some business, the reports are there, but stake and prestige of India, which is known as pharmacy of world is also important. That's why I think Government of India might have taken this stringent step to get the cough syrups tested before exporting to foreign nation. And there are some other impurities also which may occur due to reactions of nitrites, nitrates. Example is nitrosamine. Meaning thereby, we have to analyze the risk first. Assessment of the risk is must, and how it can be done. Risk-based inspections by the regulators, self-inspections by the manufacturing entity, and review by the regulatory authorities from bedside of the patient to starting material. And importantly, there are guidelines also, 𝐒𝐜𝐡𝐞𝐝 for this purpose. And even today, the major risk built is in pharmaceutical finished product, in excipients. We have to take care of excipients. It must be tested, at least for the qualitative test must be there. And with this note, I give my mic to the organizer and MOC Ms. Sinha.
Thank you.
Thank you very much.
Thank you, Manmohan sir. And these words will motivate us to work towards quality. Thank you so much for joining us, and on such a short note also. Our regards to you.
Thank you very much.
Now, moving towards the in-depth learning session on the topic of QRM today, let me quickly introduce the educator. We have with us Dr. 𝗥𝗲𝘃𝗶𝗀𝗃𝗏, founder of Pharma Mantra. Dr. Uday is a renowned expert in GXP CGMP compliance, having led global teams to achieve quality excellence and regulatory approvals. He has proven expertise in defining organizational structure for quality management and align quality function with the business objectives. He has led and spearheaded the task of developing, formulating, and implementing a global remediation action plan and strategy. He has supported organizations in steering through challenges of FDA actions, warning letters, and import alerts. He has also created self-sustaining quality management systems and prepared site and management for FDA, EU, WHO pre-qualification inspections. In past 2 decades, faced and participated in 450 plus regulatory audits, and in the corporate role, globally performed 400 plus CGMP due diligence audits. He has previous experience of working in companies like 𝗥𝗲𝘃𝗶𝘀𝗲𝗃, Ranbaxy, Gland, 𝗢𝙯𝑅, and GSK, to name a few. Welcome, Dr. Uday, and over to you to get this 500 strong audience today to get the knowledge of this topic in depth.
Thank you, Dr. Swati. Just try to help me and confirm my presentation is visible.
It says started. Yes, sir. It is visible on the screen.
Yes.
So first of all, a very good morning to all of you. I have a very pleasant morning at Nashik, Maharashtra. So it's a very cool breeze blowing here. And I appreciate FOPE, and this learning series Pulse is going to add value definitely to the national cause. And I thank Manmohanji to bring in a very interesting note to start this particular session. And it's good to see the inclusion in the part 1 for the drafted Schedule M, because I have been looking at this since 91 when I started working. And I see a lot of changes happening in the world scenario. And if you look today, the type of pharmacy of the world, which we call ourselves, and the type of documentation guidance coming in from the regulators within India is really heartening. So thank you very much, Manmohan sir. I really thank Harishji for initiating this from FOPE, and Dr. Swati, you took it forward with your team. It's really a commendable job. My friends Sanjeet, Dr. Rajiv, and Sunil, nice to see you on this particular forum today. With this, friends, I want to walk you through my thoughts today. On quality risk management. I have devised my talk on talking about the risk. So first, we'll pick it up from here, how this looks like. So if we look at the Schedule M, the Schedule M talks about the quality risk management. It's a systematic process for assessment, control, communication, and review of risk to the quality of medicinal product. It can be applied both proactively and retrospectively. Quality risk management shall ensure evaluation of the risk quality is based on scientific knowledge, experience with the process, ultimately links to the protection of the patient. So if you read it properly, friends, in this particular statements, you'll have to look in a very, very systematic way, in a different way. See, they talk about scientific knowledge means somewhere it is starting from the development. Experience with the product is right the scale-ups and the further commercial manufacturing and supplies into the market. So you get experience, you get knowledge on that particular part, and links to the protection of the patient. So patient first is the message which is coming from here. Level of effort, formality, and documentation of QRM process is commensurate with level of risk. So you need to understand the risk. Is it high, low, medium, or minimal? So that's where we are going to take you through with our experience and exposure. To start the session, we want to say what is the risk. What you see on the slide is a train coming, and there is a vehicle which is standing on the track. Now there is a hazard, there is a risk. We all understand that speed of the car can be higher than the speed of the train, but there is always a risk. What can happen? Damage to the car, accident, and this risk has to be taken care. So can a car be on a track when a train is moving? So when you look at them, no, there are some actions taken already when you look at in the real life scenario. So what is the risk? If you look at the definition which comes in here, it's combination of probability of occurrence of harm and severity of the harm. So do you see normally a train and a car together on a track? Do you read such happenings? Yes, it happens once in a blue moon. It doesn't happen on daily basis because there is an action, there is a decision which is taken, and there are some controls. So what are they? So there is a risk assessment. Probability, there is a collision can happen. A degree of severity is damage, loss of life, loss of property. How to take care of this? So safe design. So you'll make a bridge, you'll make a protection barriers, you'll have the trains, always having a gate which is manned by a guard. And then there is a traffic light, so no car or a bigger vehicle can move onto the track. But you will see there are situations where people take risk, they go below the barriers, they take the scooters, cars, and then they cross because it's going to make their journey faster. So that is the risk taken by few people when you look at this case. Now what happens? Even though it is a well-established way that there is a guard, there is a barrier, but things can go wrong. Barrier may not work, the communication signals may not reach, and still there can be an accident even though everything is in place. So having said that, friends, today I would like to bring a thought process for you that Before we get into the pharmaceutical quality risk management, let's understand the day-to-day life example. So this was on track for a railway track. Now, if we look at the scenario which is happening around us today, we all know there is a Paris Olympics happening. And in this Paris Olympics, we had few, situations where we felt bad. We were getting bronze or we were coming to the number 4. Why? If you look at the example, for example, if you look at the Olympics now, Vinesh Phogat, she gained 100 grams. Can you imagine gold, which was supposed to come definitely for us, we lost just because of the gain of 100 grams. But friends, try to understand what happens in Olympics or what happens with a athlete. They know this risk. They have to manage the weight. They have to manage the food. So though Vinesh was working very hard with her team of 4 or 5 physio, biotech, biochemists, and others helping her, she had this problem. And now we are in the arbitration court to get at least a joint silver. So there was a risk. Neeraj Chopra threw 89.34 first qualification. And in the finals, he could not throw more than 90 meters. And please understand, he tried his level best. His trainings and everything were the best. And Arshad, his friend, he threw 92.9. So this was the risk which he knew that he can miss a gold. Anyhow, he received a silver. But please understand, I'm trying to give you something which is a number which happens within the training. And now the next chance is only after 4 years. Then Mirabai in weightlifting, she started with 85 kg, very good, very nice, but she could not cross 87 or 88 kg and she lost it. Now the problem is she didn't even get a medal. Now I'm just giving few examples which are very recent examples, and the way our hockey team played, you all know that at least they got the bronze with them, and then that's where our introspection will happen. Now, going away from there now, few examples, the takeoff on touchdown of aeroplane industry, aeronautical, and the space programs, they are very perfect. And if you look at, there are very few crashes reported, though there are thousands and millions of takeoffs and landings happening. So because they work with a particular risk management plans. Birth of a child and dropping of a kid by a nurse is never heard about, because that is the personal things. You never write a wrong check. You never transfer the money wrong. Now, friends, if you just do an introspection, what happens here? It's an individual excellence, which I have been talking about. All the Olympic Games I talked about, individual excellence, except for hockey team. Then certain things you do good, and there are people who will be excelling. But in a pharmaceutical quality risk management, friends, we have to do a teamwork. That's where quality risk management in pharmaceutical happens, because there is a development team, there is a supplier management team, there is a manufacturing team, there is a testing quality team, and then there is logistic, which will take the product to the patient. And then there is a retail, and then there are distribution systems in place. So friends, I want to make a point that when we work in pharmaceuticals, there is the individual excellence which you need to do in your work, whatever work you do. And then there is a teamwork which happens right from the development till the dispatches into the market. Having said that, I make this particular context. So further, my presentation will walk you through how a team or how the things happen in various functions of pharmaceutical development, manufacturing, supply chain management. If you look at the quality definition, is degree to which a set of inherent properties of product, systems or process fulfills requirement. Risk, combination of probability, this we read earlier, of occurrence and severity. management, systemic process of assessment, control, communication, and review. Please understand, management is a area of work right from the place where the risk is happening and till the boardroom of an organization many times. So it goes through various stages. So the assessment, control, communication, risk has a product life cycle involved, and we need to understand and appreciate this particular part here. If you look at the principles of quality risk management, our WHO TRS 981 is the reference document for the Schedule M. ICH-Q9 is the reference document. Now we'll walk you through today how this looks like, or what is there in these documents which we have to implement in our manufacturing sites. There are two primary principles in QRM. One, risk to quality of medicinal product. So at any given time, you will read this term called quality and medicinal product because it's a pharmaceutical product. Should be based on, again, you see the repeat, scientific knowledge, experience, understanding of process. So please understand, there cannot be anything happening in pharmaceutical without the knowledge, without experience, and without the process knowledge. The second part here comes very clear is the patient protection. So that's very prime. So at any given time, the medicinal product desired to give a relief or a mitigation of the patient ailment. That's what is the prime requirement for making any product for the patient. It's based on the knowledge of the product or process. So friends, you will see that most of the time somewhere the words are going to be repetitive. That's called product, quality, and process. And that's where the key to the implementation of QRM. Now, when you do these activities within GMP, there is a term called, if you don't write, it's not a activity done. So you have to document. So everything which is done within a site or within a department has to be documented as QRM, and that's where is very important part. So I took some time in giving you the inputs that what's the principles and what's the expectations. This is a typical diagram which talks about a flow chart which gives the QRM process. Now, this is something which is there for last 20 years. Most of you must have seen this. On the left side, you see the risk communication. On the right side, you see the management tools. This is where it goes to the various functions which I was talking to, various areas of the manufacturing, development, and supply chain management. But when there is a risk which comes in for an assessment purpose, it may start, and then there can be an identification activity. There can be an analysis activity. There can be an evaluation activity, which may happen in a part of the site or maybe in a part of the research and development. And then it goes into the control. There will be measures which will be brought in for reducing the risk, accepting the risk, and then there will be the quality risk communication, which will happen. There will be the management tools which will be used to make it acceptable. Now we are going to talk on these things which I said here: identification, analysis, evaluation, reduction, acceptance. So further slides are going to give us some insights into the assessment part. What might go wrong? So you know, you are best, all of you either work in manufacturing, in testing, or maybe supply chain, you get the material in, or you may be in distribution. You know that whatever you do, certain things can go wrong in your work. And a systematic use of information, that can be digital information, it can be a hard copy, it can be procedures, it can be processes, which can be used and referred in a risk question or a problem description. So please read this properly, that you need to have a very good question and a very good definition of the problem which you have. Because if you do this wrong, your further steps can go wrong. So to write a problem itself is an art and is the must, because factual information is the must. Then we talk about the data which is required to look at. So you go through heaps of papers, documents, and you try to talk to many other departments, and QA takes the lead normally in the site to take care of such an activity, and then role of a team lead happens, and QA normally leads such an activity. There can be one more thought process. There's a subject matter expert can be given this responsibility, and then this can keep on varying based on the need. It can be an engineer, it can be a scientist, or it can be a development scientist, an analytical, or a process. It all depends on the problem. Then consequences. One should understand under what circumstances you may have to make hypothesis, you may have to rework on certain things, and that's where the possibilities you have to evaluate. Do the risk assessment, look at the risk. Then the evaluation part is always important. You need to look at the risk, evaluate it against the criteria which you decide, and you make it acceptable. And that's where the quantitative and qualitative part of the analysis may happen. On the risk assessment, if you look at the ISO guidance documents, you need to look at Q8. It's a development document which talks about how to develop a dosage form and what are the minimum things you should do. Then Q9 definitely talks about the quality risk management, and Q10 talks about the management responsibilities. If you read this particular thing, it talks about change controls, it talks about the various quality attributes, the CPPs, and many other things. So Q10 also makes the responsibility for the higher management and the site management. And if you look at, there is a progression from pharmaceutical development, tech transfer, commercial manufacturing, and product discontinuation. This is called the life cycle management. And there are elements or the areas which basically have to manage is the quality systems. You may have incidents, deviations, and you may have market complaints, and there can be the investigations leading to the root cause. CAPAs may be given. You may take actions. You may go through your change management system, and you may indicate to the if there is a change in process, you go back to the regulators, inform the regulatory agency that we have done this in the process, or we have changed this material, and we have taken correction. So the problem will not repeat. The whole emphasis is to avoid repeats. So that's where the risk management comes. And if you look at the enablers, there is a single statement is knowledge management. So this is very heavy on the knowledge part, and we have to always take care of this. Just try to understand that this part we have to take care. Risk control is something which we, please understand, friends, the risk can happen anytime, anywhere. I gave you an example from the Olympics. I gave you an example from the aeroplanes, aeronautical landings, takeoff. So things can go wrong. It's never a situation that things don't go wrong. Things can go, but you need to have a backup plan. You need to have cross-check. So that's why we need to understand what is the risk, what are the measures taken, and how to control and to bring down the risk on daily basis. So that's where you have to understand the reduction part of the risk. You need to understand the acceptable part also. Now, if we look at risk will never be brought down to a particular number. It will be always reduced to a particular number. Now, this is very important. Please understand that we'll learn over a period of time here, and your experience indicates that risk cannot be made zero. So that's where is the first information which we all need to note. And so comes the control, that there will be always a residual risk. There will be always something which will crop up as a problem in future. Now, if you look at the type of quality management systems we operate within the sites, we always have an investigation, we have a root cause, and we have been trained to give a correction. which normally is a immediate and a reactive activity. Then you go to the next step, you say, I'll give a corrective action, and again, it's reactive to that particular activity or that particular incidence or non-conformance. And then you try to give something long-term, and then you try to eliminate the root causes. Now, in correction, you immediately address within few hours and you take the next decision because you have to move forward into the manufacturing or testing or delivery processes. Actions have to be initiated to take care of the next batches. So normally correction can happen for a particular batch or an incident, and then corrective actions can happen for a number of batches or something. And preventive action is the proactive things, friends. And this is a prevention part, which will ensure there is no repeat. And that will be called preventive action. And you'll have to check the effectiveness that it should make potential problems known. So you'll have to work on this. Reduction. Now, what's risk reduction? We say always. So we have to understand that risk is always there. Risk may be increased or decreased because of many factors. It can be internal factors, it can be external factors, and you need to understand that on daily basis on a packaging line, on a manufacturing suite maybe, or a filling line, or you can say even within testing in microbiology or maybe laboratories, things can go wrong. So the whole thing in reduction is try to identify and look at the occurrences within your systems and procedures, look at the detection possibilities, and that's where the key lies in identifying. So you need to record. Suppose you don't record, friends, the risk, incident, deviation, market complaints are not recorded in the batch records, or if they are not written as per the QMS, there is the risk which we are saying that we are ignoring it. And you will not record the occurrence, and so detection will not be there. So there is always a risk of high failures in such a system. Risk acceptance is a decision making, and this decision making will always be a team activity because a lot of subject matter experts, they come, sit, and they take a call on a particular decision. Now, residual risk, as I said, will always be there, and then acceptable level has to be understood case by case basis. So there can be a lot of debate, discussions, but you can take a call. You can record, this is my acceptance criteria, and that's why we are moving forward, or we are not moving forward. And that's the way you work. You may re-decide on this after a particular review, and there is a possibility your risk assessment, you may revise and you may say, this we missed, and it can happen. Some information comes after some time, and then you realize that our assessment was biased and it had some corrections to be done. You can do that activity once again. So please understand, when you accept a risk, you do that based on information, and information, if added afterwards, changes your decision. Feel free to do that. Never be shy to change the decision once you have more information in place. Risk review is an activity which leads to new knowledge, new experience. It's very, typically a learning phase. Whenever you look at any problems, we have some case studies for you today here. I'll walk you through. And it's really interesting when you look at the problems, and at times it's very simple cause, but we realize that it has happened, it was neglected, it was not understood. The impact was not understood. So the impact and the original risk assessment, it is the experience which can come to an individual after a lot of working, lot of review activities, inspections, audits, change control. So that's where the knowledge comes. And in any activity within the systems, there is always the ongoing quality management. You do APQRs on a yearly basis. That is annual product quality review. You do your trend analysis for each and every activities. And then you have this activity called review happening for each and every batch, each and every parameter. And that's where the knowledge is getting recorded and it is getting enhanced. So that's where the new knowledge and experience comes in a review. Failure mode analysis is a tool. This helps you to assess, manage, and reduce the failures. So I'll just walk you through how this particular system works. So it's a quantitative characterization of failure. It's comprised of assigning probabilities in three factors like occurrence, detection, and severity. So these are three terms which we will be talking in this particular section here. And you need to understand that this can be done with a number which appears as a RPM. Okay, so that becomes the risk priority number. And based on that, you can say the risk is high or low. That's the way you can do this activity on daily basis. This is a 𝐒𝐜𝐡𝐞𝐝 method when you do the investigations in the site. For example, here there is a process control contamination for quality and safety requirements. So what you look at is the 5M, 4M in investigations at your end. So this picture depicts how the man part is looked at training, then they have to comply to the procedure, and the practices may be as per the procedures. And when you talk to the operators or the supervisors, you realize there are gaps in the procedures. So you need to do the gap assessment here. Methods and instructions, there are test methods which are validated. Then there are parameters which have to be looked at, like pH, conductivity, temperature. And if there is a variance and if there is something not complying, you need to have a investigation. You need to have an incident recorded, and that is must. It has to be a part of the batch records. If there is an automation, you need to have the printouts in place. And similarly, when you look at materials, you have a lot of things which come into the plant. It can be raw material, the water, the air, and many other things. And in the machine part, you have the process equipments, you have the different various unit processes involved where the equipments are used, but each batch when done by a particular set of people or a particular material from a vendor can behave differently. So this is just an idea for all of you to look at the various aspects of an investigation. Occurrence is normally having numbering 1, 2, 3, 4, 5, and 1 is when the occurrence is very low. For example, if filling is happening and in the filling room something falls down, it is very rare scenario that someone will pick it and put it on the line again. Normally they will be trained to remove the vial, ampule, or maybe the pre-filled syringes and keep it separate. It won't go back to the line. There is a very low probability of such a aseptic behavior happening. So it is detectable and it is possible. So the failure rate can be low. So occurrence can be low, failure rate can be low. But suppose if something is happening and which is having a high probability of failure, the number given will be 5. Now we'll tell you how this is done with an example. Then comes severity. Severity is normally to make this particular situation as that particular activity incident is a serious thing, or is it going to be less serious? It will depend on the severity classification. Now, if we look at this table, if the product quality is not affected, it can be rated low. And if it is going to be affected, that is the severity can be treated as high. That's so you look at the critical product parameters, you look for the critical quality attributes, and this is within the batch records or maybe the COAs or maybe the test reports, which gives you the specifications high and low. So that's where you look at and you take a call, and then there can be corrections. Detection. Detection is something which, you need to understand that certain things can be detected and certain things cannot be detected. So to help the detection, maybe the visual inspection, for example, a manual process done by a few people for the tablets, injectables. So when you go for replace a manual inspection by camera inspection, probably it enhances the part of the inspection. And further IPQA check or a supervisory check by the production people indicates how this camera system is working. But please try to understand, a camera also needs the teaching. It has to learn, and then only it has to be implemented as per the SOP with the defined challenge tests, which are written as a part of the batch record You have to indicate this was done, it was complied, and then camera was put into use. So that's the way you start your day. So it has a probability of detection. So in that scenario, you can say my probability with a machine is very high to identify a black spot, maybe on a tablet, maybe a cracked tablet, a chip tablet. It's good. And even manual system can be good, but my camera is going to be much better. That's the way you rate your lines where there is a manual system or there is a automation. And if you look at the rating part here, again here, one thing a bit different here, if the rating here changes, if the detection is high, it is just other way, please understand, the number given is 1. And the detection is not good. There is a tough scenario. For example, microbial contamination. You can't see if a product is sterile or non-sterile. You don't know the CFU colonies unless you incubate by eyes. So on the day one, this is not reported. So that is very tough to identify. So it is such type of risks are on a higher side. So anything which goes wrong in the process or procedural gaps will lead to high risk, because the detection is going to be on a different scale. You will not understand until tested. So please try to understand detection. When it is having probability, the ranking is 1. But when there is no possibility of detecting certain things, it will be on a 5, scale of 5. Now, what is an RPN? RPN is occurrence, severity, detection. You have to put these numbers. And these numbers can be between 1 to 5, this can be 1 to 10, and there can be some other numbering system which is applicable. But if you look at the theory part and if you look at the application, you have to multiply the occurrence, severity, detection, and you get a number. That's called the RPN number. And if you look at 10, 10, 10, the number can be like that. And if it is 5, 5, 5, the number will be equivalent coming on a higher or lower side. Now, let's look at the decision tree. If you have this information in place, how do you look at this activity? So in case of the contamination, which we were looking up in the fishbone diagram, so what happens here? If it is a known pathogen, then what? You can take a call. We can say it is yes. Okay. And if the microbial contamination isolates are first time, and if it is yes, do subsequent steps in the process have the capability to eliminate or reduce the risk? There is a risk which it comes when it is yes here. But suppose is the microbial contamination isolated to the first chromatographic step? This is talking about a biotech system where there is an upstream contamination. The risk is no. It is said no, it is not having any information. Then again, there is a risk. And then when you look at this particular thing, microbial contamination, known pathogen, if yes, you have to go to the next one, bioburden levels. Then you look at the microbial contaminants in the toxins, endotoxins, other things. And then you look for the toxins and still there is a risk. But there are times when you can take from this decision, when there comes no, there is no potential product risk and you can take a call. So that's where the risk acceptance or further investigation decisions taken here. I will give an example where we will put some numbers to the problem. These are few steps in the FMEA. That the steps are put some more time on this. We'll go to the section where we'll like to look at the numbers here. This is an example I would like to take from the step 1, 2, 3 here. Review the process, determine failure and mode, and determine potential risk. I'm taking it here on the top. So just for your information, I have written on the top 1, 2, 3. So if you look at the drying process in an organization, there is a setup, there is a possibility of contamination, and the gowning can be an issue. The insufficient cleaning can be an issue. Drying, when you start, there can be still a contamination, damage to the filter, degradation of the product. Then maintain temperature, the LOD And because in a drying process, you normally work on the moisture content. And these are the potential risk here. Now, if you look at the numbers, severity of contamination, 3. It happens here for the 𝐷𝐶𝐻 gown. Occurrence, it is 50-50 probably on the scale of 10, and it remains there. And then detection, people have not gone properly, everything, it is visible, answer is yes. So it is detectable as 𝐷𝐶𝐻 gown, but can you look at the contamination microbiology? So the detection level is on a higher side, you cannot, because it's very tough for even chemical contamination to be seen when the work is happening. So please try to understand here detection levels are low. The number, if you look at going 8, 6, 3, this is the area of concern. Occurrences is something to do with your procedural parts and the documentation which you did in the site. And severity is something which happens to a product where you have to take a decision to accept a batch or throw a batch. So if you look at this SOD, this is the numbering system, and then you can understand on day-to-day basis how to look at this also. So please make it an easy way to use this in your manufacturing testing areas. So I'll go to the next slide, which talks about now the number. If you look at calculate the RPN, if you multiply 3, 5, 8, it comes 120. Then 7, 2, 8 comes 112. Now, if you look at wherever the detectability was an issue coming up, the number in this particular slide, okay, there can be a situation for you where severity can be high, the occurrence can be on a very higher side. But in this case, I am taking you through is the detectability is not possible for contamination. Look at, and that number goes more than 126. This is something which you have to look at. And the other numbers like 63, 40, 8, 27, 5, they are again the numbers which come up, but then you have to focus in this red band. What you do next is 8, 9, 10 steps. You look at the modes, you look at the eliminations, you look at the recalculation after the mitigation. So to avoid this particular contamination on the gown of the operator for contamination by the body or by hair fall or something, use long gloves and goggles so you protect the product. So you brought down the number which is 120 RPN with a severity 3, it remains same, occurrence reduces 2, and the detectability still is not possible. It remains 8, but still your RPN comes down. So please understand the actions have been taken and this will reduce the risk. So the same way there is a cleaning procedure, occurrence reduces, severity is still on a higher side and detection has increased. Maybe there is a test method involved, maybe there is a cleaning and visual inspection and some samples as rinse and swab to support. Change maintenance period. So look at this, the number still remains on a higher side for the inlet filter, but it was 126, it has been brought down, still remains 84. So this is the way you have to look at the RPN and take a decision on the calculating. Try to work on this, which I say on the bottom of the slide is when RPN is over 100, take decisions, take actions when severity is over 5. This is severity part over 5 and critical parameters after taking action. So let's look at this. This is a table which talks about the activity which is called the risk assessment. Now, if you look at this is the whole table done, I'll walk you through only the important part which I made red here is pre-mixing. The number you see is 18. Then there is 27. Then there is 36, 36, and max is 36 and 17, and minimum is 3. There is an action, there is a risk reduction plan, and then it has been brought down to 6. Now, if you look at risk reduction, it is 18 minus 6 is 12. It is 27, that is in the pre-mixing granulation. It is 6, it has been brought down to, and the reduction is 21. So friends, see the numbers. So it is quantitative techniques. It can be definitively shown that there is a number and it is now reduced by 12, 21, 32, 32. This is the way you have to present, you have to work on this. Now, there is a possibility of one more activity when you take it up into the categorization part. You can take it a dashboard of the type of risk assessments you have been doing. So you can make some level of risk within the assessment, and it can be X1, X2, X3, X4, X5. X5 remains still severe if you have to still work on this, and X1 is acceptable, no action. Training can be for X2, cost effective and selective controls X3, and X4 can be control irrespective of cost, immediate change of process. You may have to take a decision to stop the activity here. Now, quality risk can happen from system risk. It can happen from organization, facility, process, and packaging. Now, if you look at this table, which I showed you in the start, it has this particular thing, which is a very standard ICH requirement. And at site, you work actually. You do the investigations, you do the FEMA, HACCP, FTA, and you do root cause assessment, you do corrective actions, you do the risk assessment, and you have a lot of documentation. Friends, this is the way any regulator will like to understand. So sequencing at site has to be somehow managed the way this green table shows. Now, this is something interesting, which I said that the issues can come from anywhere. The issues can be within the sites, outside the site, external, internal. All these things can come to you. It can be at the site. It can be at different levels. And then you have to get into risk assessment, identify, you have to go for the control, you have to go for the review. And there is a good activity which happens is you have to review it into a monthly site quality council meeting. And there has to be a project update to risk mitigation. And there can be some, indicators, green, yellow, orange, which can be used for identifying and making a report for the management and at the site. So this is what is the example where we make it a management reporting system. I want to take you through, I have some time here. I kept it purposefully. I'll walk you through two quickly case studies here. This is a fungal contamination study which I want to show you. This involves recall of multiple batches. Background. In response to adverse observation, firm recalls 45 batches for fungal contamination. Quality investigation revealed source of contamination was an ingredient in the product called DCP, 𝐷𝐸𝐴𝐶 calcium. Non-compliance was observed during the audit by the inspectors, and they found that there is a TYMC not more than 100 CFU per tablet mold and yeast, and it was increasing, and the product went into the market. And when they looked at the contaminant, it came from environment, and it was typically Penicillium, Aspergillus, and Botricolium. Now, if we look at the investigations which was shown and understood by the site, it's a common environment contaminant, which is there as a part of their wild isolates from the site. Now, quality site investigation brought in this particular ingredient as the causative factor or the source. The use of DCP, if we look at, and the impacted batches, this was something which they had to do within the site. And which now, if we look at the material, so there was a vendor investigation. And when the vendor was investigated, surprisingly, it was observed that vendor makes DCP and the larger consumption is other than pharmaceuticals. It goes into paste, it goes into confectionaries, it goes into paints, it goes into diverse type of industry. And then it was realized that there was a air suction, there was a system where that was not having any filtration when the drying of the DCP was done. So the correction was that there was a drying step put in. The work with the vendor was done, and then there was a particular quality to be monitored at CFU level for the input material. But the company had to recall this particular 45 batches. Now, what is the risk? There was a contamination risk of a particular yeast or mold. Answer is yes, but it is very difficult to tell when you are releasing the batches, because who is going to take this and how this will become a patient risk is very tough to imagine when you're releasing the batch. What is the impact? Impact is the evidence when you receive or you get a source. It can impact a single batch or there can be multiple batches. So there is a cost which gets involved here. There is a company's reputation also gets involved. So you need to have that thing in your mind when you're doing the impact assessment on one batch or multiple batches. Lessons learned is usage of DCP and the vendor qualification activity. And please understand that if pharmaceutical industry needs a particular grade, you need to make it a part of the purchase order, make it a part of the specification, make a vendor audit, make a vendor qualification, and then allow it to be used in that manner. This very simple example is the cough syrup problem which happened, and the propylene glycol and DEG. So if you all understand what has happened with the cough syrup was nothing but someone had contaminated glycol used for the cough syrup which has DG and DEG. This is the second example which I take it from the process part. This is a risk associated with fungal spores. So fungal spores, please try to understand, is not only a single colony appearing, but it's a full colony available. So some fungal spores growing on the surface of clean rooms. The background is the case is about a sterile product manufacturing facility which had routine excursions in the clean rooms for fungal spores, and it was identified as Aspergillus brasiliensis. The levels were on a higher side, going to the action levels. Now, this was in the change room, and the excursions were observed and recorded. Investigation was focused into the filling room. They were near to the point of fill, point where the vials were getting exposed or they were getting stoppered, and they were trying to look at all the values on the those areas, and they were coming okay. The only issues were coming in the change rooms. Now, the full focus went on to the drain and the areas near the filling line. And operator, if they looked at the downing, it was all coming okay, no mold or nothing was there. So the issue was that from where these spores and other things are coming then. Now, when they did further investigation with a third party or maybe a subject matter experts, and what they found that the active air samples of the operators entering the room tended to show high counts when they were entering. Rest, it was coming all fine. Then they said, let's look at the cartwheels, look at the other things, and the swabs and many other things were done around the entry, exits, and the filling room. And then their focus was then coming to the opening and closing of the door near the entry part. And then they realized on the door there was a kick panel, a plate which was put in where the operator, you were trained to kick it and enter there. So that was the way they used to push with the feet inside the door, and then they used to enter. So this particular thing, when they realized there is a plate, they removed the plate SS, and then they found that there is a black mold around this. And when they took the samples, it was the colony. So the risk to the patient, a sterile product going out with mold, yes, it is a patient risk. How is the impact? Fungal spores outbreaks are increasing, and this is getting reported in many cases in ophthalmics, if you look at, in ointments, if you look at, even into the various type of injectables. So this is a common problem reported if you read worldwide today. And this is something, you know, spores will start growing in humidity and warm temperature, or it may remain dormant. So this particular type of contamination is not acceptable. It is used in surgeries, or if it is used in someone with a diseased condition. So instead of correcting the problem, it will increase the problem. So that's where the impact. Fungal contamination is leading to sterility failure, excursions, loss of production. What is the lesson learned here? So the lesson learned here is that this kick plate which was there installed, it was a part of this particular process, always the design of the building equipment and facility, and no one realized that this is having the contamination behind this within the clean room. So this was a missed area, And the second things which they commonly use, which goes in or out or which is missed, can be bags, boxes, intervention equipment, cards, shoe covers, markers, scribbles, pallets, glassware, trolleys, and jacks. So I think these are some things which we need to look at as a QRM benefits, friends. Help reduce overall cost, decision making in planning, promotes quality through increased efficiency, knowledge transfer. You look at the continuous process, it's not focused on a particular day, event. No, it's a continuous process. It cannot be reduced to a particular activity. It's a knowledge updation, it's experience which you gain, and it's a process on a monthly basis as we recommended. One has to keep on working on this and keep on discussing, debating, and looking at the risk communication part and then reduction part. Second important part during this communication is understand the process, understand the criticality, and bring on all the rationales. Helps provide a rationale for not spending time on low risk activity. You'll realize over a period of time, you'll know the risk by this RBN numbers, by the risk management. So the focus, energy, and the money shall be spent on the high risk activities. So this is the way you can help the management, and the journey will continue. So that's where I stop my presentation. Thank you so much for your patient hearing.
We would not like you to stop, because that was a fabulous masterclass that you took for all of us today. It was really, really awesome with case studies, with simple examples. The way you have designed the presentation, the way you delivered, sir, hats off to you. A wonderful masterclass. Thanks to you for bringing this sort of learning to these sessions. Harisha, would you like to speak something on the quality of the session?
Normally, if you look at I being an entrepreneur and not involved into day-to-day affairs of quality control and quality assurance, the risk, the QRM and the risk is normally a kind of, I would say, a kind of a boring subject for me. And because you need to really concentrate on this subject. And when I was hearing 𝗥𝗲𝐃𝐶𝐴 and his presentation, it was like a story. It was a story, and the kind of and so much of, I think, today what I learned on QRM is, I think, I have never learned in my whole life. And this has been excellent, and so happy that we have 𝗥𝗲𝐃𝐶𝐴 with you. The kind of case study is so simple, and especially those tabular things. So it is, how do we quantify the risks involved, and how do we manage those risks? I think there is a kind of a very eye-opener kind of a thing. Thank you, 𝗥𝗲𝐃𝐶𝐴.
Yeah. So, sir, you heard Harisha. Thank you, Sanjeet sir, also, because he has been there all along for designing the learning sessions, designing how we should be presenting and all. Sanjeet sir, would you like to speak a few words, and then I will move forward?
Thank you, Dr. Swati, and thank you, Dr. Uday. Uday and me, we have been working actually from more than 30 years now, and then we have discussed many areas where there are practical implications of risk management for day-to-day kind of issues, whether it is complaints, product recalls, investigations. But I think Uday made it very simple to understand for people, and his explanation of the basic risks, starting with the Paris Olympics to the train station, and that makes things easier to understand. And that's what we all have to do in managing the quality of our product. We have to make it simple for our people so that they can understand and they can apply. I'm sure that this will go a long way in understanding this subject. This is just the beginning. But as Uday said, you have to apply it very regularly for a continuous improvement in your operations. Thank you, Dr. Uday.
Thank you, Dr. Sanjeet. Dr. Uday, thank you. And please stay with us for the panel.
Swati, if you allow me a word.
Yes, sir.
Thank you. Thank you so much. I think it was a pleasure to speak on this topic. And this is a very difficult topic, as said by Harish bhai. So, and please understand, this is my version 6. And my friend Sanjeet made me work at least 15 days on this. So just put it on.
We avoided the risk of not presenting the up to code.
Not only that, he ensured that he gives me a call 10:15, and then Dr. 𝐒𝐜𝐡𝐴𝐬𝐴 also was with me at a session at 10 o'clock. So both of them, they ensured that I come here in time. So there were two more people behind me too. So what I'm trying to say, it's not individual now. It's a teamwork for you also.
Right. Yeah. Thank you, Dr. Uday, and please stay with us for the panel discussion. We will start the panel discussion. Questions will be directly asked from the participants. So participants, please pour in your questions. But before that, we also have with us today Mr. 𝗥𝗲𝘃𝗶𝘀𝗶 𝗦𝗏, the Chief Executive Officer of 𝐿SSSDC with us, Life Sciences Sector Skill Development Council. Welcome, Mr. 𝗥𝗲𝘃. And let me now, we have something new for you today, for the audiences. You have learned on this topic from the subject matter expert. So Satish, can we have the questions? Today we are going to take a test for the audience also. So can we have the questions quickly? Please answer 10 questions. They will be flashed on your screens. Use laptop to answer these questions. Yes.
Yes, I'm launching the first one.
Please participate. We will give 30 seconds to each of these questions. Question number 1, quality risk management is a systematic process for the. Please choose your answers. Yes, more participation. We have more than 500 people live. So please.
Yeah, at least 50-60% will be highly appreciated. Please participate. Yes, you have gone through the session.
The questions are not out of syllabus. They are from the presentation itself. So please participate. Yes, so we have crossed 300, Satish.
So I'm ending it and then showing the result to everyone. So this is the result.
And the correct answer for this one is the second option: assessment, control, communication, and review of risk to the quality of the medicinal product. Let us go to the next question.
So yeah, stopping it. We are going to the second one.
The question is, quality risk management can be applied retrospectively, both proactively and retrospectively, both proactively and concurrently, and both actively and passively. Yeah.
Oh, wow. Majority.
I think it was on the first or the second or third slide. I also saw the answer.
Great, great. Keep participating. So once we cross 300, we'll stop it. Show the result. Wow, it was quick. So now, yeah, we are ending it and then showing the result. So here is the result.
And your maximum
Just
is right.
Is it visible? The result is visible. Swati, is the result visible?
Not yet, Satish.
Why is it not showing the result?
So the correct answer is both proactively and retrospectively, and 77% have answered it.
So just give me a few seconds so that I can put the screenshot in the group so that it's there. Yeah.
Let us move towards the third question.
Third one. Let's move to the third one. So this is the third one. Just a sec. I think it is facing some issue. Let me relaunch.
So we will take up 5 questions, Satish, and we will keep
Yeah.
5 questions towards the end of the session when we are ending
Yes.
the whole session, right? So we are over to next third question. Please launch the
Just
question.
a sec. No, yeah, now it is showing. I think there is some problem in Zoom window. I'm clicking on the launch. It is launching, but not showing on the screen.
So
Let me try the fourth one. No, there is some issue.
So let us keep the questions for the
Let's continue and then we can do the
Yes.
sessions.
Yeah, yeah, there is
Yes.
some issue in the technical issue is going on.
That's the risk when you go live.
yes. Sir.
Yeah. So usually, sir, doesn't happen. Today it's causing.
That's the risk.
Risk.
Yeah,
Yes.
that is the
Satish,
That's
issue.
called residual risk.
Yeah.
That's the residual risk. Yes.
So let us, Satish, now play the video. This is a strategic timeout, and we want to play the video of the sponsor for the session. And after that, we are moving towards the participants Q&A. So Satish, can we have the video of 𝗤𝗏𝗃𝗏, who are the sponsors for the session? Thank you, 𝗏𝗏𝗏, for sponsoring the session. Moving towards the panel discussion, let us all welcome the panelists. Today, we have with us Dr. 𝗥𝗲𝗃𝗶 J, Deputy Drugs Controller, Regional Office, Bengaluru. About him, he is a seasoned pharmaceutical professional with a PhD from JSS Academy of Higher Education and Research, Mysore, and extensive experience in drug regulation, having served as a drugs inspector, assistant drugs controller, and currently as deputy drugs controller for the past 3 years. He has a proven track record in ensuring drug quality and safety. He has successfully led numerous initiatives, including combating spurious drugs, enforcing drug regulations, and managing critical public health issues like AMR, medical oxygen shortages, and tuberculosis eradication. Recognized for exceptional contributions, Dr. 𝗥𝗲𝗃 was awarded the prestigious National Award as Best Drugs Controller Officer in 2023. Welcome, Dr. 𝗥𝗲𝗃. The second panelist today we have with us is Dr. Rajiv Desai. Dr. Rajiv Desai, most of us know him, a PhD MBA with over 38 years of experience. He is a renowned leader in pharmaceutical quality and regulation. He is currently the Senior Technical Advisor at the Indian Pharmaceutical Alliance, IPA. Dr. Desai boasts of an impressive career spanning India and Switzerland. He has held leadership positions at Lupin, Dr. Reddy's, Mylan, and other prominent companies, managing quality across all aspects of drug development and manufacturing. His expertise extends to regulatory affairs, project management, and technology transfers. Recognized for his excellence, Dr. Desai has received prestigious awards, served on expert committees, and actively contributes to the advancement of pharmaceutical industry. He was also a member of US Pharmacopoeia Expert Committee from 2010 to 2015 and currently supporting USP workshops and seminars as an invited speaker for topics in pharma industry. He is a member of Indian Pharmacopoeia Expert Committee and also nominated by the Ministry of Chemicals and Fertilizers, Department of Pharmaceuticals, Government of India as member of the Board of Governors for the NIPER for 3 years since November 2022. Dr. Rajiv, welcome. And the third panelist we have today is Ms. Mala Sivaramakrishnan. I think in this session only we have the pleasure of hosting a female leader. So welcome, ma'am, with over 3 decades of rich experience in the global pharmaceutical industry. Ms. Mala is a seasoned quality professional who has made significant contributions to some of the world's most renowned pharma corporates. Currently serving as the director and head of site quality at Cipla, Ms. Mala's leadership and expertise spans across multiple areas, including quality control, regulatory affairs, compliance, operations, and vendor management. Her extensive experience also includes key leadership roles at Hikal as General Manager of Quality and Regulatory and at Biocon as Head of Regulatory for the US market. Her commitment to quality is not just limited to her professional achievements. She has been a passionate advocate for education and coaching. She has chaired syllabus curation processes for industry academia programs and has been recognized as a woman leader by the Karnataka Drugs Manufacturers Association. Moreover, has leadership in guiding teams through numerous regulatory inspections and over 600 customer audits globally. It speaks volumes about her dedication and expertise. And beyond her professional contributions, Ms. Mala is deeply committed to social causes, actively volunteering in CSR activities too. Welcome, Ms. Mala. And we also will have with us Mr. Harish Jain, President FOPE, and Dr. Sanjeet Singh Lamba, Managing Partner, Trillium Consulting, to take over this session of question and answers with participants. So over to you, Harisha. Please unmute requesting all the panelists to please unmute. Yes.
Yeah, thank you, Dr. Swati. I think in addition, we also have the speaker as well, who will be part of the panel discussion, 𝗢𝗏𝗏𝗏𝗏 and also not to forget our chief guest Manmohan Taneja ji. I think some of the questions probably I would put it to him as well to answer. And thank you very much for all the panelists for joining in and also all the audience for joining in. Stay put up. I think I'm flooded with the number of questions. I don't know whether we'll be able to finish all the questions. But then what I will be doing is probably during the presentation and the chief guest remarks, whatever questions are already answered, I will not take it up. And I will give priority to the questions which are relevant to today's topic. And that will be my modus operandi of conducting this question and answer. Now, I think the very first question The woman first in this panelist, I think the very first question goes to Mrs. Mala. How do we apply QRM in development of contamination control strategy?
Okay. So thank you, Dr. Arish and Dr. Swati and the panelists here for giving this opportunity for the session. Well, it's a very good question, I would say. Sometime back in August 2022, we had the Annex EU GMP Annex 1, which specifically mentioned on this contamination control strategy. So if we have to do the risk assessment related to this, then we need to understand 3 to 4 basic parameters, which is nothing but the process, what is employed, because Dr. Uday already mentioned about the scientific knowledge. So we should know about the process, what you're going to employ, what type of processes are involved as a unit operation, and the personal monitoring that should be there, which is very, very important because the contamination could be from person or the process what we are employing. And the qualification process, the qualification with respect to your equipment, as well as the different processes and the unit operations that we do, and how we simulate it on a regular basis. So on these factors, if you can identify the risk, okay, what are the risks which potential risk which exist, and what are the contributory factors for these risks, and of which circumstances these potential risks could be possible, that we should know, based on which we can know the severity and the current control measures. What are the different current control measures we have where the severity can be reduced? So having known that, we can know what is the detectability level of the risk which is there. Okay. Followed by the contributory factors. What are the contributory factors with respect to handling the different processes or the unit operations which can give rise to the potential risk that will be known, basis which you can derive an action plan so that the risk can be mitigated or it can be nullified. I'm just summarizing it so that I don't take more time on this question. So it's a very, very large topic because we should see how to reduce the potential contamination while we are doing the different processes and mitigate the risk.
Okay. Thank you. I think the next question I'll put to Dr. Bhaskaran, the Deputy Tax Controller, Bangalore. A little out of context, but I thought let me still take it up. In the revised Schedule M, point number 14, subpoint 14.16, it says that appropriate procedures or measures are in place to ensure the identity of the contents of each container of starting material from bulk containers from which sample have been drawn shall be identified. The question is, what we understand about identity, the question is in 3 parts. What do we understand about the identity of the material? Can we omit the identification of the material when we perform identity of the material? I don't know, but the question is little, I don't know, not easy to understand. If material has more than one identification, in that case, composite pool sample identification, is it sufficient?
Thank you, Dr. Harish, and for bringing me onto this platform. It's a very important question, which in fact everybody is, because here what we see is the risk is directly associated. The risk factor is directly related to the quality of the product, and it is directly related to the patient, where who needs the quality product. And as it says, 14.6, it is there should be an appropriate procedures or measures to ensure the identity of the content of each container. The 3 parts accordingly what has been asked, I think so. The first was
what we should understand about the identity of the material at the very first part.
What we should understand about the identity of the material. Yeah, of course, because API comes in bulk in different though the batch is same, we have a number of containers. As a regulatory point of view, every container has to be identified individually, is a mandatory requirement. And that goes without saying that yes, it has to be identified. Identification, particularly, every container has to be identified. And the next question is what, Mr. Harish?
If I would skip directly to the third question in that is, if the material has more than one identification, for example, paracetamol, in the Indian Pharmacopoeia, there are 2 or 3 different identification tests are given. In that case, composite pool sample identification, is it sufficient?
Okay. See here, what happens is, as a regulatory point of view, we start with right from, if you take 14, the materials to be purchased, and if you get into that, 14.7 says the purchase of the starting material is an important operation and shall be purchased only. Later on, it says that it should be purchased only from an approved supplier, and if possible, directly from the producer, is the content goes on. But here, because this is an important risk, which is directly to start with, the risk is involved. Every identity is an important factor. So every product, though it is in a different containers, every active ingredient, every container has to be identified. Though if it has got 3 or 4 tests also, each has to be identified separately. Then the other test can be performed on it can be combined together and performed. So it's better we identify all the tests. Identification says that if it is 1 or 2 tests also, identification has to be done on individual basis, is what I feel, what the regulatory system says.
Okay, I think we got the point that identification test has to be done on separately of each of those containers, and composite we can do all the other tests.
Yes.
Anyway,
Yes.
Going ahead to the next question, I'll put that to our today's speaker. Is FMECA tool mandatory to do risk assessment, or shall we proceed with ISO 9001:2015 risk matrix to do the risk assessment? This is to the 𝗥𝗲𝘃𝗶𝘀𝗲𝗃.
Yeah, I think response to this question is, see, as per your procedure and SOP, you are allowed to perform the risk assessment. So there is nothing called mandatory this activity. Even if you look at the schedule and the way it is written down, it says risk assessment, risk management. So it says you refer to so and so. So it's left to the manufacturer, it's left to the SME, what you are comfortable with, what is easy for you guys to work on, please pick it up. And FMEA was just an example which we brought in. The fishbone was just an example we brought in. There are various techniques you can use. Absolutely okay.
Okay. Going to the next question, I'll put that to Dr. Rajiv Desai. Is detectability mandatory to consider for conducting risk assessments?
That's true, Harishji. Thank you for this question and thanks for this forum. Basically, risk assessment is to identify how you can mitigate them and how your processes can be made safer. So it's essential that any parameter that we identify as part of the risk assessment should be definitely quantified, because as Dr. Uday mentioned in this presentation also, we have a concept called as RPN. RPN takes care about all the parameters, and it actually tells you how good or how easy is the process. What's important is that when you talk about this quality risk management, I know the companies who are working in the regulated markets, this has been a regular feature for them. But it's important that they do it for the benefit of the product, for the consistency of the product. And at times they really forget that it is not only for their regulatory consideration for just to manage the guideline, but it's for the product also. So it's always better to take care of the risk, understand the risk, and mitigate it in a much better manner. What we find is, as Dr. Uday mentioned about the RPN, there's a categorization of RPNs, low, medium, and high. Most of the times, everybody would get every number as low. And this has been quite a common feature that no matter what risk is involved, you always get a low. And then they say there's nothing to be done. It's not possible. And at the same time, your risk mitigation should be linked to your failures, as you mentioned very rightly about so many events happening, the out of specifications, the market complaints, the stability studies. I mean, there cannot be a simple thing as just mitigating or getting a low value. So it's important that try and get the maximum risk in that, try to mitigate in a better manner so that you don't end up in a failure in your future products.
I think I'll put that next question again to you, Dr. Desai. I will probably combine the next two questions. How many risk assessment tools are currently available in the guidelines? And what's the difference between harm, hazard, and risk?
Oh, see, as we have heard, see, there are several tools. Tools are just to tell you what should be used. It can be a fishbone diagram, it can be FMEA, but you should not be restricted to the tools that you should use. As we said, that a risk can be identified with common sense also. So first use your common sense, then try to look at the structured way of doing it. But it's important that you identify the risk in a faster manner. These tools are just trying to make it in a structured manner to ensure that you come out with a better mitigation plan, and that's the whole idea. The guidelines would talk about maybe in a couple of months' time or years' time, you'll get another tool of identification. So don't get restricted to the tools. It's a common sense that should prevail, and the risk that you have identified based on your experience and based on the others' mistake. I mean, in the pharma industry, you don't mistake and say that मैंने गलती किया तो मैंने मेरी गलती से सीखा. आप दूसरों की गलती से सीखो, and that is what is important. And not try to make sure that you end up in the same issue, in your processes. As you asked about the contamination, it is a major concern for us, and it should not be repeated in our manufacturing processes. Your next question was about trying to understand the differences. Yeah, there's a difference.
Harm, hazard, and risk.
Yeah, so harm, hazard, and risk, I mean, it all depends on how it affects your product quality, how it affects your patients. Patient is, at the center of the whole manufacturing. It's not just any kind of industry. Everybody should realize the importance of the seriousness of the business that we are into, and make sure that we realize it ethically in the practices. So it is important.
I'll put that next question to Sanjeet Singh ji. Can you please unmute yourself, sir?
Yes, sure.
Whether QRM can be documented reactively for deviation or out of specifications?
Yeah, good question. I think we had one question here in the polls also, that QRM can be applied. Better way is to apply proactively rather than reactively. You have to I said the situation should change now from 𝗏 to 𝗏, in my opinion, because you should work more on a preventive action and then the corrective action first. And mostly what happens is as a reactive mode, when actually the situation is like this, that you already have a problem, you already have an incident which has happened, it has to be applied concurrently in that situation, because you know that there is a problem and you have to really assess what is the risk on this particular issue, whether it is a contamination, whether it is a DT failure, or dissolution failure, stability failure. So it has to be applied both proactively as well as concurrently as well as retrospectively. So this has to be applied in all the stages.
I think
one more thing I just wanted to say is, in my opinion, over the last many years, I have seen the most important decision the management and the executives and the people around quality and manufacturing they have to take is, how do you design the detection? This was one of the questions. In my opinion, detection is most important. That if you can reduce the probability of occurrence, that's very good. Many times severity we cannot reduce. Severity remains the same because a patient is at risk. What we can do and what is in our hands is to really raise that detection. Even if there is a failure, I have a detection in place so that the bad product cannot go into the market. That's the whole purpose of a risk assessment, that the bad product, the bad quality product should not reach the patient in any situation. So that's the way. So if you can automate a process, if you can put a camera inspection system, take the control from a human being or a procedural SOP control or a document control to an automated control, as Japanese say, Pokayoke system, which means mistake proofing. Even if somebody tries to commit a mistake, the system should not allow you to commit that mistake. So that's the whole philosophy you should base your quality risk management on.
I think great. I think as you rightly put, it's better to be proactive rather than reactive. I think moving further, we might see some kind of a predictive risk risk identification tools also coming in. So I think that will be, I think some of the experts on this panel as well as in the audience should be working on it. Then the next question is, what is the correct approach, whether it is qualitative or quantitative for the RPN number? I will put that to our speaker, Uday Kumar sir. What is the correct approach, whether it is qualitative or quantitative for the RPN number?
Friends, again, I always go with one message. One is that what is your SOP talking about? Because you have to comply to your written systems in place. Now, qualitative is something, which you take action without going into documentation, and you have to take a call, which is called immediate. For example, if there is a problem, pilot is having a problem with his communication system, then he has some other way to resolve that. He takes a decision based on that. Similarly, when you look at an operator in the clean room area, and if he has a problem on the machine, so rather than calling an engineering person, normally we train them to resolve that particular part, because it is a risk to call a person from outside and the intervention to be caused. So I'm trying to give you an example where the numbering he will not calculate. The person is trained to take action, and then you will find that some of the operators, they are expert on machine, that is the filling line, or maybe the tunnel, or maybe the capping and the bottling areas, even labeling and packaging. So you see, qualitative is one activity which happens immediately just as corrective and can be recorded. But when you go for the number, the way Dr. Rajiv answered, that how that detectability, how that occurrence and severity, where Dr. Sanjeev said, then when severity is on a higher side, you need to have a number. So my suggestion here, qualitative is the common sense, and you record it, you cannot miss it, and quantify it. So that's the way you have to work. So qualitative will always be followed through with a quantitative assessment.
I think the extension of that question would be, what is the scientific rationale for assigning numbers for RPN? Can we fix RPN in-house methods?
Yes. If you look at the way we started discussing severity, now we had a chat very clearly given thoughts that what is severity? It is a patient protection. Even the number one cannot be ignored. Let's understand this. But we have a correction and we can take a call. But if you look at severity, which is 5, there is a vaccine or there is an injectable having a contamination of a pathogen, cannot move out. So that's where the high severity. So anything which is severe for a patient perspective has to be investigated. So number 1, 2, 3, 4, 5 is just a rating number. It can be 1, 2, 3, 4, 10 also, or it can be high, medium, low. But severity has to be attended. So no issue. Now the occurrence part means if there is a multiple occurrence at a particular site, like many times the inspectors, they come in, they say, last time also I saw this time also I saw this, occurrence and repeats need to be taken care. So there was I think a message from the panelist, my fellow panelists, that occurrence has to be brought down. That's called the risk control, which I discussed. Now the detectability part, if there is a manual activity and there is a automation, but you ignore the printouts, you ignore the signals, and the trend is not documented, that's a risk. So number many times becomes deceptive. I many times use the term that the numbers are deceptive. Qualitatively, you may look foolish at times, but please understand, you take decisions on your common sense, and secondly, the factual information. So at no point of time, the factual information can be ignored. So numbering 1 to 5, 1 to 10, and then taking a decision has to be SOP driven. So I always recommend, why? Because whenever the inspections happen, whenever you do investigations, you have to answer the question based on your documentation, and that documentation has to give a very clear picture under what circumstances severity, occurrence, and detectability has to be measured, or how it is to be investigated for a root cause.
I go to the next question. How to make preventive action for human error? I thought, let me answer myself. Probably what we have is human error prevention. And what we normally see is when you do the investigation, the whole of the human error, it actually boils down to your system failure. I think so. If your investigation and if you can fix up your system failure, then you can definitely reduce your human error. And we in FOPE, we also run a program. It's an offline program called as Human Error Reduction, which we did in Hyderabad a couple of weeks back, and which we also plan to do across India. It's a very important tool when we look at the investigation of the human error reduction. It actually boils down to your system failure. Correct your system and you can reduce your human errors. The next question I'll put to Mrs. Mala. Can we prepare one QRM of plant starting from store to dispatch? Is it possible?
Harish, I just want to touch upon the human error before I go to this question, if you are okay with it.
Yeah. More the merrier.
So what I feel in the human error is basically many times you have your processes, many times you have your procedures in place, but because action did not get executed as per the procedure, then also the human error will happen. So it will be leading to the competency of the person and understanding of the person, whether that is at fault. If that is at fault, how do we derive it? So it is better that every company has some checklist around the procedure that they have, so that we will know whether it is related to the competency of the person the error occurred, or the understanding of the procedure. Because complex procedures, when we write, then also the human error could happen. So I think that is one of the factors to understand the procedures before they start executing it. Now, coming back to the actual question, what you put, right from dispensing up to the final, maybe the dispatch risk assessment being written. So normally in industry, these are called as a product risk assessment, okay? Where end to end you write a risk assessment for your product, right from dispensing activity. So what are the different risks which are involved in the dispensing activity, right from receipt of the material, whether it is an approved vendor you're sourcing the material, and what kind of truck the material is coming in, whether the truck is clean, and if it is not clean, what control measures you're having it. So right from there, it starts. I would say that it starts from security, because they have to let the right truck inside your factory to get the material unloaded. So from there, the risk starts. The transportation of the truck, whether the required MSDS is available for the transit of the material in case of any spillage, how do we handle this material, whom to be contacted, and whether there is a TREM card, that is, what are the contacts while the truck is getting transited to the company. So even your vendor engagement is very important, I would say, when you're talking about a product risk assessment. Your vendor is aware of the risk during the transit of the material. So from there it starts, and to different components we can break this risk assessment into, so that every function is involved while doing this risk assessment, and every stakeholder is involved to know the product risk and how it can indirectly impact on the product quality and patient safety. So one is a dispensing part, then the quality control comes into picture, whether the specifications are well defined, the sampling process is well defined, any source of contamination during the sampling process or during the receipt of the material, then dispensing of the material to the production after the batch is completed its testing. Then again, the control measure or the process during the processing of the batch. Then comes if there is a non-compliance during the manufacturing, like deviation or any changes, unanticipated changes, how do you mitigate those risks? That can be built in proactively, as Dr. Sanjit mentioned. followed by the release processes and the dispatch process. So there can be a chain of activity integrated into the risk assessment, which we call it as a product risk, instead of the individual risk assessments.
Okay. Next question, I think I will put it to Sanjit Singh ji. I think this is one of the question which all the MSMEs always we have that kind of a question. How to do the risk-based audit? Internally, how do we do the risk-based audit?
I think it's a very good question. Thank you, 𝐿𝐴𝐴𝐴. Actually, if you look at your overall day-to-day kind of an operations, there is risk involved at every stage. When we even start in the morning from home to go to office, there is a risk involved. There can be an accident on the way. There may be a time frame. I may have to reach in 15 minutes, but there may be some kind of a damage on the road, which is not allowing me to reach my office in time. So there is a risk. Similarly, if you see that for every product or a process, there are some inherent risks. Some risks are covered by companies as a part of their risk assessment, and some risks are not covered. So you really need to look at, if you are a multi-product facility, you have to categorize and classify your products into high risk, medium risk, and low risk. High risk products would be those products which are injectables, followed by ophthalmics, and followed by what damage they can do to the humans if there is a sudden contamination found in a product. Whereas if you see on a derma kind of a product is a ointment which is applied on the skin and not in the eye. If it is applied in the eye, again, it's a high risk area. But if it's a derma product on the skin, the product risk is little lower than as compared to others. So first is a classification. Second area is between the classification, you look at some products which are very sensitive to moisture, sensitive to compression pressures, sensitive to environment, sensitive to many other areas. So if there are sensitive to the processing conditions, you have to classify it under oral solid. You say my 𝗥𝗲𝐃𝐿𝐴𝐵 product, for example, is a high risk product. So you have to define your high frequency for auditing those products, those processes, which require more attention from you, because just that the risk involved in those product processing is high. Or finally, even those product per se, like injectables, are high. So you have to really prioritize the audits in such a way so that you give priority to high risk items first, followed by the medium and then the low risk. So accordingly, you can make your inspection and audit strategy based on this.
Thank you. I think the next question I'll put to both the regulators who are there on the panel. I think the thing is a very interesting question, very general question, which will, we all of us face. How do we prepare for risk-based inspections?
Yes, well,
Here, I think the CDSCO in collaboration with the state regulators are conducting risk-based inspections across the country for almost last 1 year. So I'm just wondering, how do we prepare for risk-based inspections? I think start with the senior pro, the Manmohan Taneja ji.
yes, Mr. Singh has already given this answer. Depending upon the risk of the product, you should prepare your same. If you are stringently doing self-audit, role of regulator will be minimized. We also conduct risk-based inspection on the basis of the risk of that particular unit. There are criteria for NSQ drugs reported from different laboratories, different state regulators, and even by WHO. On that basis, risk-based inspection is conducted. If a unit is failing time and again in their product quality, risk base is conducted. If you, as Mr. Singh told, you should analyze the risk involved in your products, high risk products and low risk products.
Okay. Bhaskaran sir.
Yes. As a regulatory point of view, it's very clear as the discussions is going on from morning. QRM in place brings everything into place. The SOPs, whatever has to be. See, we see the documentation is the main criteria. Whatever is being implemented has to be documented. Documentation is what is the important. Every part of it, every risk or assessment or whatever it is to be done, control has to be done, has to be documented. This documentation is what the risk inspections will further carry on. So whatever has to be done, whatever will be done, whatever the control or the review has to be done, has to be documented. Documentation is very important, is what we're looking for. So I think so this should help.
Yeah, correct. I think any failure is not an end in itself. I think probably what is the kind of activity you do after that failure.
Yes.
I think that is more important. Whether what is the kind of investigation you have done, what is the failure assessment you have done, and what is the preventive action you have taken. I think that is extreme. And for your systems, what you have put in place,
Yes.
quality system and a lot of other things. I think that is what is important. Is what it is coming out from both of your views.
Mr. Harish, one more small extension of this is, see, this QRM should not stop within your facility. The QRM has to be extended until the product, until the patient. See, what happens is the NSQ, whatever has been a product which has been declared of NSQ or whatever NSQ, if it's declared for NSQ, it's the duty of that it should be within the part of the QRM so that every NSQ drug. See, as a regulatory point of view, we always go behind the NSQ drug. But the QRM also should speak about the withdrawal of the product. Not even a single product, single tablet should be consumed after the product is declared as not a standard quality. That should be the process which is to be involved, is what I feel.
Correct. I'll put the next question to 𝗥𝗲𝐃𝐿 ji. What's the difference between risk and impact assessment?
Thank you. I think it's a good distinguishing question. What is the risk and what is the impact? So if friends, you look at the risk is on the track. When I was there, I said risk is the risk is to the driver who is in the engine driver. The risk is to the person who sits into the car. And there are many other people around that where the impact can be there. So this is the risk on the track. Now, what is the impact? Impact, you heard the direct impact onto the engine and the car and the people around. So that's where you look at from the high level. Now you come to the process part where I give the example. There was a risk of DCP getting contaminated. Now, this particular detectability was low. Please understand, you cannot identify each and every bag and then there is a problem unless you test. So the risk in many times in pharmaceutical manufacturing is when you test as per the specifications, then only it will be identified. And suppose if an organization is doing skip testing, what I mean by skip testing is you don't perform a test for all consignment. You say every 5th batch, every 10th batch, I look at the pathogens. There is a probability in between you have missed those particular consignments where there was a possibility or a probability of contamination. So that's the risk. And now what is the impact? Impact is simple, friends. Recall, I said by Dr. Bhaskaran that when you find something NSQ that is not of standard quality, regulators expect you to recall the product, the impacted batch, and all the other batches where this material has been used. So it's a huge financial impact. It has a huge quality impact. And definitely patient risk is high because there is a pathogen contamination. So I think that's the way one has to look at the risk and then they have to look at the impact.
I think the next question I'll put to Dr. Rajiv Desai. Very interesting question. How to conduct risk assessment during development of solid dosage form finished product at the early stage of selection of starting materials?
A very good question, Harish ji. Because what happens is when we talk about these guidelines, especially quality risk management, we look at that, okay, it's been published by ICH and it's been also referred by the regulatory agencies during the inspection. But many times we lose the track that these guidelines should also be percolated down at your initial phase of your product development. And it's important that the people at the development, people who are into R&D, should also realize that it's not a one-time activity that they do the research and push the product to push the technology to the manufacturing, because it's a life cycle management that's very important. And in our kind of scenario where we are into generics, it's always a plan to push the product as fast as possible and minimize the number of experiments and development that we do in the product technology. And it's very important to understand what has happened around the world. I mean, they should also, R&D, I understand we respect the R&D technical expertise on the chemistries, on the formulation technology that they carry. But it's very important to understand what can go wrong, because at the manufacturing side, we do not have technical expertise the way that the R&D folks would have, who would understand the nitty-gritties of the technology. We need to develop processes which are easily understood, which are easily manageable at the site, and do not involve any kind of very complexities. Of course, the complexities are increasing, but we should also support them with the technologies that we can bring in at the manufacturing. But it's important that at every stage of the development, whether use of the raw material, whether use of the processes, developing a methodology of analysis, developing a SOP or a batch record is so very important. Every step has to be looked at, that what can go wrong and who can make mistake. I mean, when you talk about the human reduction and human errors that are there, it's basically not been able to give the kind of support that requires in the manufacturing, and we start blaming the people. But we should also retrospectively look at what can be done in a simplified manner right in the development phase, which is so very important for the product to go on a regular basis. Commercialization is important, consistency is important, and developing at a very hasty manner the company will definitely pay the price for that they would not be able to, and they will have this product going back and forth in the R&D to get it right. And risk assessment is all about getting the process in a very robust manner, which should not be just left to the manufacturing side, should also percolate back to the R&D folks.
The next question is how to revise the risk assessment. I think probably I would say is through the, we'll have, I think, go through that process of change control. And we'll have a session separately on the change control. Definitely, I think any change you do, it has to be through a change control with the concept of risk assessment and everything. I think I'll go to that. I'll put that question when that session comes in. Again, to Dr. Rajiv Desai, any guidelines of QRM for bioequivalence studies?
Oh, there are guidelines for the bioequivalence studies as well. But what is important is that the guidelines on the clinical studies and the bioequivalence studies that we have are undergoing a major change now. There are talks about minimizing the animal studies, minimizing and coming out with different technologies. And we should look at whether these studies have been done in-house or whether they have been outsourced, and whether the outsourced studies are really in line with the requirement, or is it done just for the commercial benefit of the product, and to make sure that these are to be evaluated. We talked about internal audits. It's also important that the audits should be done at the place where these studies are done, and making sure that these guidelines are to be followed. There are guidelines where we go through them and ensure that they are done. I think there's a big impetus coming from the Indian regulatory agencies also, and it's a good time good thing to know that even in India, now we are getting this into our system, and a lot of importance is given to these guidelines. We have to understand the technical involvement, the technical advancements coming up in the clinical studies, the bioequivalence studies, which are coming up, to make sure that we understand the requirements and we take it forward with a much better understanding of the needs of the pharma development and the commercialization.
I think next is a bit of a breaker. I think It's a more of a comment by one Judy. Awesome session, sir. It was really interesting, giving insight on current quality tools, how to implement. Looking forward to have more session like you, like this. Congratulate all the panelists and speakers for you are the rightful owners of this comment. And the next question is again I'll put to 𝗥𝗲𝗃𝗶𝘀𝗲𝗱 presently, pharma industries are following 𝐼CH-Q9. I would like to know what's the revised Schedule M is different from 𝐼CH-Q9.
I think the response to this is that in the disclaimer and within the gadget notification, if we read it properly, it has been mentioned quality risk management as a part one of the notification. And in that, the reference documents are coming up is WHO and ISO. So what happens is that it's a requirement from the regulators. They will not recommend what you should do because it's an understanding. So one has to go and look into the best practices followed within the world. And so the reference mentioned is 2 year, that is WHO and ISO. Even in my presentation, I picked up the same. But you are free to pick up, as I said, some options which you feel is better and good to follow, where you can quantify and qualitatively perform a risk assessment. So that's the way one should look at. And please understand, as I was talking, that this activity inside daily, there is a drift in manufacturing. There is always a drift in the material received. There is always a drift which happens in your critical process parameters and critical quality attributes, and you measure on daily basis as a certificate of analysis and batch records. So you need to identify those incidents. You need to look at those particular drifts, and that's where comes your quality management system called incidents, out of specification, out of trends, and market complaints. And there can be many more in preventive maintenance or somewhere else where you can document such things. So once you identify such things, you need to do an investigation. So investigation, risk assessment, root cause, and preventing the repeat to happen in the plant is the responsibility of the manufacturer. So whenever a regulator will come to you, they put all the onus on the manufacturer. How you perform is not the recommendation. It will come from a regulator. The performance is as per your SOPs, and your SOPs are called procedures. Procedures says do this, do this, do this. And if there is a gap which is observed that you are not recording, you are not documenting, and you are going forward, OS not recorded, OOT not understood, investigations treated as minor, as said by co-panelists that the severity is mentioned low, and then the RPN number is brought down to say that I don't need an investigation. This type of activities are no-no, because the inspector who comes, they are enough knowledgeable. The inspectors which come within our own system in country, they are absolutely understanding what's happening. So guys, please understand, there is no one here who is going to accept such an investigation which says severity low, detection high, occurrence low. No OS, no deviation, no investigation is not the trend, not acceptable. So please follow the systems which you want, but follow the SOP, because if you don't follow SOP, you have done a good job, still it will be objected by the regulator, because if you don't comply to the SOPs, that's a major observation you'll find. So it has to be understood from that perspective. Thank you so much.
The next question I'll put to Mrs. Mala. If the risk assessment report already approved and we need to incorporate some additional assessment or design control or some more information, how to revise it?
So Harish, I'm taking up this question along with the previous one, which you had parked it for another session. So I'll combine that and this and take it together. So when, as Dr. Uday was mentioning, the development of a procedure for risk assessment is very, very important. Because once you have your procedure, in the procedure, you have to clearly write at what frequency you will be revisiting your risk assessments, which are already prepared. So this can be into two categories. One is that you have a risk assessment for your product, and then you have a deviation or repeated occurrences of deviation or some complaints are there. then you can revisit your original risk assessment to see that what are those factors which contributed for this departure. Okay, so even though the risk assessment could be approved, but you can revisit to check what additional control measures you need to incorporate so that the non-conformities are not repeated. That is one thing. So where you can keep a risk assessment number when you're doing it, it can be part of your procedure. A risk assessment number can be assigned and you sign off the risk assessment. And again, if a problem occurs or you feel that this risk assessment need additional information, you can go to the next version number, change your risk assessment, add the factors which were missing in the previous risk assessment, and then you can sign off. And then again, have a periodic review, maybe once in a year or once in 3 years, whatever is your company procedure, and revisit it such that there are no non-conformities in terms of deviations or incidents or complaints, and whatever control measures you put is workable and it is giving you a consistent product.
Okay, thank you. I think the next question I'll put to Bhaskaran sir. In case of technology transfer of existing product batches from one location to another, shall we conduct process validation? And also, is it mandatory to wait for stability study outcome before release into the market? You are on mute, sir.
Can you repeat it? Can you repeat it? Yeah.
I'll repeat the question.
Yeah,
In case of technology transfer of existing product batches from one location to another, we shall conduct process validation. But is it mandatory to wait for stability study report before it is released into the market?
Such, I don't think so. It's required. Stability studies is not required. Process validation should be well enough to carry on.
Yeah. There is a letter from CDSU also in this regard.
Okay, so no need to conduct the stability
Yeah.
studies.
Yeah.
Okay, because the US FDA guideline says that even in case of transfer from one site to another, stability, 6 months stability is for 3 batches is mandatory.
As per Indian regulations, even CDSC or DCJ has a circular regarding this.
Okay, good.
Harish, before we move on, we have now 15 minutes past the dedicated time. We'll take up more questions also. And
we have, according to me, I have another 40 questions also.
No problem.
So what I'm trying to do is I'm keeping the questions which are not relevant to the topic.
Yes. One important question, Harisha, I saw in questions, and this is directed at CDSCO. I would just like to ask this. Foreign regulators like US FDA, EU GMP, etc. do post the inspection outcomes in their website. Will CDSCO have such any plans to post the inspection reports in their website based on risk-based inspections for the benefit of manufacturers? This is coming to regulators, Dr. Bhaskaran.
Bhaskaran, sir.
Yeah. Can you please?
Like foreign regulators like US FDA, EU GMP post the inspection outcomes on their websites. Will CDSCO have any such plans to post the inspection outcomes in their website based on the risk-based inspections that are going on for the benefit of manufacturers?
But you should ask the
Oh, no.
You are talking about the CDSCO inspections.
Yes.
Joint inspections, in fact, WHO inspection, joint inspections is well aware of it. In fact, right now there is nothing like, we don't have anything like that which has been posted on the website. Dr. Manmohan sir, please share your thoughts about it.
Okay. So, Harish, over to you for the next question.
I think what my assessment will go in that respect is there are different laws. Now, apart from the drug control laws, there are other laws are there which are different in India as well as in the United States, whether it's a transparency or whether it's a Right to Information Act, or what in US, the equivalent version is, I think, Freedom of Information Act. So something like that. So there are different regulations are there. So whether the posting of those inspection reports on the CDSCO website violate those laws in our country, I think that will play a major role in whether the CDSCO publishes our inspection reports on those websites or not. I think that would be my take on it. Okay. What is the difference? What is the difference between generic? Again, it's out of context. What is the difference between generic medicine, ethical medicine? Is there any difference of manufacturing process, quality? Pharma companies are highlighting ethical products are better than generic products available in the market. Again, I'll put this to both the regulators.
See, we look in, when it comes to the quality of the product, we look in whether we don't look in whether it's a branded product or a generic product. The quality standards are the same. The specification speaks, according to the specification, standards remain the same. The procedures remain the same. The facility remains the same. There is no deviation in this. So we look in the same way, and the testing, the quality product will remain the same. This is what we're looking for.
I think Manmohanji, sir.
Yes, absolutely. He is absolutely right, correct.
I think what we should understand is in India, unlike other commodities, there is no first quality or second quality. It's only first quality. So I think that people, so whether it's a generic or branded, if you look at paracetamol tablet IP, it is paracetamol tablet IP complying with all the specifications of not only pharmacopoeia, but also the GMP standards as notified under Schedule M. I think this is what we should understand.
The specification remains the same. We have only one standard book, Indian Pharmacopoeia. Yeah. So it has to comply to that.
Okay. I'll put the next question to Dr. Rajiv Desai. Again, very interesting question looking into MSME point of view. What is the easiest way to apply QRM? Implement QRM.
Easiest way?
Easiest way to implement QRM.
So I'll just take back to some of the discussion which happened a couple of minutes back on the internal audits. See, try and make sure that give empowerment to your internal auditing team, which is so very important. Nobody can tell what are the finer details of your process. You have enough experts within your team. Try to identify the best people who can be part of the internal auditing team. They can identify the process. They can know what can go wrong or what are the issues of the similar kind of processes. I think that's the easiest way. You can get people from outside, but getting people from outside, they are on the learning curve. They will not understand the finer details of your process. But identifying your cross-functional team, as we call it, to conduct an internal audit, you'll be able to identify more risk. Try to make sure their processes have become more streamlined, and that's what the base. I mean, I would say the easiest way to do a quality risk assessment.
Yeah, I think every company should understand. Every person should understand the product and process is owned by that particular individual or the company. So correct. Everything, whether it is a process, material, equipment, people, competition, it's only you are the best judge. And based on that, within your organization, what is the risk involved? What you need to identify the risk within your organization. That risk might be different for my company or the other neighbor's company. I think this is what, so based on, you should have also, I think the whole Schedule M GMP is based on the science. And what is the science we use to identify that risk and also mitigate those risks. This is what would be, I think, the outcome of today's session. I think another interesting question is how to avoid biased risk assessment, as many times it is just a formality. I think anybody can volunteer.
I think I would say the same thing, that as long as you do not pin down the risk around the issue, you should be able to take it very positively, identify and make sure that you have a corrective actions and preventive actions well defined. And that is what we say. I mean, looking at your past data, looking at the data or the events from your other manufacturers, looking at the observations which have been cited, this will give you a much better way of identifying your risk and making sure that your processes are made robust. And that's how it should work. I mean, rather than just, as you said, looking at the RPN number and just putting down the number at a very low level and saying that, oh, I don't have a risk in my process, is not a good way of the QRM.
I think in view of the paucity of the time, Yeah, Sanjit
I
Singh.
just
Yeah, you
want to add, it's very rightly mentioned by Dr. Rajiv. So here, I think we should also hear from the shop floor people because they also have a good experience of the day-to-day challenges. So we have to involve them into this so they understand that there is a process of risk assessment and how it could impact on the different unit operations they do and how it could impact on the product quality. So we have to make sensitize those people as well in both analytical as well as in the process or in the development, because in current situation, it doesn't start from development. So from there, it has to start, as Dr. Rajiv rightly pointed out a couple of minutes back. So once we give this knowledge and sharing to everybody as in PQS, okay, I think we will be successful with this.
I can just add here that risk assessment is not like something written on stone. It's always an evolving document. Taneja ji also mentioned that there are so many issues. The most common issue that we are facing is on the nitrosamines. We never knew this 5 years back, but we started looked at our processes and saw what we have been doing. I mean, we have been making the products for the last 10, 20 years, and now we opened up our R&D and also tried to understand where the issue came up. Having done on the nitrosamine, now we are on NDSRI. Again, we opened back and went back to our drawing boards and looked at what risks are involved. So never shy out of saying that why it was not done earlier. It can always be done whenever you feel like, and there's nothing like a hard and fast risk assessment. You should always be open to ensure that your processes are always on your toes.
I think great thing. Like I think our the QRM as a tool can be applied to every activity in our organization, and that becomes not only a kind of a many people think that the QRM is a more of a hindrance, but I feel it's a more of a tool to solve your problems.
It's an enabler.
So I think I'll take one final question in view of the paucity of the time. We are almost approaching 1 o'clock. Against our 12:30 we are supposed to end. I'll put to both the regulators both the question: how can a self-inspection play an important role in pharmaceutical industry? First to Bhaskaran sir, and then the final word with Manmohan Taneja ji.
This is the industry person has to reply to this. Very, very, very important. Okay, when you are perfect at your home, there is the external person. I mean, as a regulator, we cannot point out anything about it. When you say that, because we both are equally responsible for the quality of the product. See, ultimately, the patient is the quality product has to reach the patient. Whether you as a manufacturer or as a regulator, the main criteria, main important, this one is like we need to give a quality product to the consumer. Is what is our role. So it's not the QRM inspections, all this is a second part of it. You should have an attitude to give a good quality product to the consumer is what should imbibe in yourselves. So this is what I feel.
Manmohan ji?
You are in the mute, Manmohan ji.
Sir, you have to unmute yourself. Taneja ji, you have to unmute yourself. Sound nahi aa rahi hai.
I do agree with Mr. Bhaskaran, and I have earlier also answered this earlier question. If we are doing risk assessment and all at our own, we are following the Schedule M, and regularly we are doing self-audit controls and all. There is no need of even much more stringent inspections. The severity of the inspections will reduce automatically. Inspections are, we select the units on the basis of the previous archives. If there are no major or critical shortcomings or contraventions during the process or in data integrity, in documentations, there is no need of even inspections, what I feel.
I think slowly
I mean to say regulatory inspections.
I think like global practices, our own country is also going from inspection Raj to compliance Raj. So once you do the compliance, I think there is no need for a kind of not being confident about facing inspections. So I think that is what I would conclude. Over to you, Dr. Swati. I think I will
Harish
end
bhai, if you allow me one point here.
Yes, sir. Yes, sir.
Just one point after listening to this, which I think I want to make it very clear and very recommending that human behavior plays a role in all these activities. And when I look at this particular scenario, the fear of failure is something which is going to be a face-off for a development scientist, a manufacturing chemist, or a testing analyst. So somehow we have to bring in the possibilities and policies where we allow them to tell the mistakes and allow the mistakes to be rewarded. So in industry, what's happening, the success is the numbers. If you gain, you have complied, you have done this much of numbers, but there is no reward for identifying a problem and giving a solution to that. So I think in the system of human resource and human behavior, my work has given me a scenario where there is a cognitive bias which happens in an investigation. A person who does an investigation, if he's from R&D, he will put it into manufacturing. Manufacturing chap puts it into somewhere else, and then it goes into a merry-go-round. So the question many times is it only self-inspection or internal, or there has to be a third-party involvement? I think there has to be a corporate cell, or there has to be a compliance cell, which is said to be done. So which gives a third eye view. So I think for a implementation, which question was there, if you have to do a good QRM in implementation from the management, there has to be a support and there has to be a compliance team, which takes care of the single site or various sites and which does it unbiased, because there is always a bias which comes into investigations to protect their own interests. So that is probably a human factor I wanted to chip in with.
Correct.
Thank you.
Yes.
Over to you, Dr. Swati. I think there are many more questions. How do we handle? I think, as usual, we take all the questions. Couple of days are not sufficient.
You're right, Arisha. We will compile all these questions and try to answer them through various agencies, various experts also. That will do. Thank you, Dr. Uday, for a befitting remark towards the end of this session. And yes, I'll now invite to do a vote of thanks. But before that, just give me a moment to show what is upcoming for the Pulse series for all of us here. So as you can see on the screen, we have done 3 sessions on general overview and implementation plan, pharmaceutical quality system, and quality risk management. There will be a filler session and we are planning for this. We will give the information that how, if the session is scheduled or not. After that, you have from session 4 till session 25, a lot of important topics coming up. These will be those sessions which will be, very important for you to comply for revised schedule M. And these will, the sessions will be packaged now. We will not be going live from session 4. These will be closed groups, and the registration and the payment and the certification will happen for all these sessions together now. So the next session is upcoming, qualification and validation on 8th, and all the information will flow to you through our WhatsApp groups, through your mail, and many more channels through our social media. And now I will invite Mr. Arjun Kapoor, if he's here. Yes, Mr. Arjun Kapoor, Additional General Secretary of FOPE, to give the closing remarks and vote of thanks.
Yeah, thank you, Dr. Swati. A very good, visible, and interesting morning converted to afternoon to all. I think I'm audible.
Yes, audible. A bit of network issue.
I'm audible, right?
Yes, Arjun.
Yes. Okay. I think there is not much to be said now after such an extensive exhaustive, illustrative, easy to understand, and interesting session that we just concluded. Would like to start by thanking and congratulating both PharmaState and FOPE for completion of another high-value, full-power knowledge session on this much-coveted Pulse series. Special thanks to our sponsors for today's swim trial, and not to mention our chief guest, Dr. 𝗥𝗲𝗃𝗶 𝗦𝗫𝗍, who inaugurated this session, spoke on quality risk management, the topic for today, how it is the responsibility of not only the regulators, but also the industry captains hand in hand. Topic is quality risk management. Our eminent speaker, Dr. 𝗥𝗲𝗃𝗶 sir, you have spoken on quality risk management has to be done both proactively and retrospectively, with particularly an example of car and training, these kind of examples which got us very much interested throughout the session. I'm sure we still have a good gathering of participants, good number of people, glued to this session because of the kind of examples and the kind of easy to understand presentation that you just gave on QRM, that essentially contains points like product quality and patient protection, and the steps: identification, analysis, evaluation, FMEA, risk assessment, decision tree, and the process related SOD numbers, severity, probability, and detection of failure, calculation of RPN, and topping it up with some real on-the-ground case studies. A wonderful masterclass indeed, sir. Accept my sincere thank you on the same. Followed by an array of eminent expert panelists, Dr. Bhaskaran, Dr. Rajiv Desai, Ms. Mala, Dr. Sanjeet Lamba, coordinated by Harish sir, and Dr. Swati. The way they have effectively answered the real-time problem-based question of our audience by giving extensive solution-based approach towards the same. This is the first time that we included the, right from the first, we have included the questions of the real-time questions of the people who are attending the webinar. And I think it had gone really in a very interesting manner. We didn't want to conclude because of the paucity of the time. I think it's a very interesting session that we have just included. Last but not the least, a big thanks to the audience who have graced the session on a beautiful Sunday morning and added value to Pulse series of excellence and to themselves as well. Thank you, and over to you, Dr. Swati. That's it from our side. And I wish you the best, and the best to the Pulse series that is coming.
Sure. Thank you. Thank you, Arjun. But participants, we faced a failure in the polls, right? So there is a plan for it that we have devised. You all are in WhatsApp group, and this poll will be taken up in the WhatsApp groups. The rest of the questions will be posted there. We will not spare you. You will not spare the test of learning. So you have to participate and give us the right answers in the WhatsApp group already there for the sessions. So towards the end, I wish you all happy Independence Day. You can see in our backgrounds, we try to incorporate somehow the flag of India, and 15th August is now 4 days from now. Happy Independence Day to you all. But my biggest thanks to all the speakers and panelists and participants today who joined us, contributed to this session and made it a session. I think I'm going to get a lot of requests for recording of this session. So we will try our best.
Dr. Swati, not only the recording, I think the presentation of Dr. 𝗥𝗲𝗃𝗶, I think our speaker, already I'm getting lots of messages. Please share his presentation.
Yes, that's true.
Yes. So that is the collaborative effort that we put in. And I hope we set up those standards which we will accept for us in medicines as per the quality. We will take such medicines which are generated with all the quality systems in place. So let us respect that, and we move towards the next session on validation on 8th September. Please join us. All the information will flow to you. Thank you, Harish sir. Thank you, Sanjeet sir. Thank you, 𝗥𝗲𝗃𝗶 sir, for being here as chief guest, and Uday sir for speaking on the topic, which was of most importance given the inspections that are going on. Thank you, Dr. Bhaskaran and Dr. Rajiv and Ms. Mala for being here with us. Thank you, Arjun, for being there.
I think special thanks to Sri Manmohan Taneja ji, who has been there from the start till the end. And his short address, I think, put his points very clear to the audience. What is the expectations of the regulator and what is the role of a regulator in ensuring the quality of the products. And also thanks to Dr. Bhaskaran sir. Again, his answer to the questions have been crisp and he's been up to the point. I think thank you both the regulators for sparing your valuable time, and FOPE and PSA is always will be indebted to you. Thank you.
Thank you very much.
Thank
sir.
you.
Thank you.
Wishing you all happy Independence Day. Let us meet
Thank you. Same to
in
you all.
September. Yeah. We are closing off now. Give us your feedback. That is the most important. We tried to make this session as per your feedback, more time for Q&A. And let us see what feedback we receive this time.
Thank you all.
Thank you, everyone. We are
Thank
ending the
you.
session now.